Hello everyone, today we will discuss the part 61 of the important notes for biochemistry. Urea cycle disorders. So first of all, the hyperammonemia type 1, that is deficiency of the carbamyl phosphate synthetase 1 enzyme causing accumulation of the ammonia. Now next is the hyperammonemia type 2 disease, and over here there is deficiency of the ornithine transcarbamylase enzyme resulting into accumulation of the ammonia, uridine 5' monophosphate, uridine monophosphate and the uric acid. Citrullinemia type 1 disease, over here there is deficiency of the argininosuccinate synthetase enzyme causing accumulation of the ammonia and the citrulline. Now next is the argininosuccinic aciduria. There is deficiency of the argininosuccinate lyase enzyme causing accumulation of the ammonia and the argininosuccinic acid. Hyperargininemia, there is deficiency of the arginase enzyme causing accumulation of the ammonia and the arginine. Citrullinemia type 2 disease, over here there is deficiency of the citrine, that is the aspartate glutamate transporter causing accumulation of the citrulline. Triple H syndrome, it is an autosomal recessive disorder due to deficiency of the ornithine transporter causing hyperammonemia, hyperornithinemia and the homocitrulinuria. In the glycolysis pathway, the rate-limiting enzyme is the phosphofructokinase 1 enzyme with the regulators or we can say enhancers as AMP and the fructose 2,6 biphosphate, and the inhibitors are the ATP and the citrate. While in the tricarboxylic acid cycle or the Krebs cycle or the citric acid cycle, the rate-limiting enzyme are 3 enzymes depending upon various conditions including the citrate synthase, isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase, with the enhancers or the regulators as the ADP and inhibitors are the ATP and the NADH. In the glycogenesis pathway, the rate-limiting enzyme is the glycogen synthase with the enhancers and the regulators as glucose 6-phosphate and the insulin, and the inhibitors are the epinephrine and the glucagon. In the fatty acid oxidation process, the rate-limiting enzyme is the carnitine acyltransferase 1 with the enhancer or regulator as the malonyl-CoA, which enhances the particular oxidation. In the fatty acid synthesis pathway, the rate-limiting enzyme is the acetyl-CoA carboxylase with the enhancers or regulators as the insulin and the citrate, and inhibitors are the glucagon and the palmitoyl-CoA. In the cholesterol synthesis process, the rate-limiting enzyme is the HMG-CoA reductase with the enhancer or regulator as the insulin, thyroxine, and the inhibitors are the glucagon, cholesterol, mevalonate, statins, and the bile acids. Now next is the ketone body synthesis. So over here the rate-limiting enzyme is the HMG-CoA synthase. So remember the difference. In the cholesterol synthesis pathway, the rate-limiting enzyme is the HMG-CoA reductase, while in the ketone body synthesis, the rate-limiting enzyme is the HMG-CoA synthase. Over here, the regulator or the enhancers are the glucagon and the inhibitors are the insulin and the malonyl-CoA. In the HMT shunt pathway, the rate-limiting enzyme is the glucose 6-phosphate dehydrogenase with the enhancers as the NADP and inhibitor as the NADPH. In the glycogenolysis pathway, the rate-limiting enzyme is the glycogen phosphorylase with the enhancers or the regulators as the epinephrine, glucagon and the AMP, while the inhibitors are the glucose 6-phosphate, insulin and the ATP. Now in the gluconeogenesis pathway, the rate-limiting enzymes are the pyruvate carboxylase, PEP-CK, that means the phosphenol pyruvate carboxykinase, fructose 1,6 biphosphatase with the regulators or enhancers as the citrate, and the inhibitors are the AMP and the fructose 2,6 biphosphate. In the bile acid synthesis, the rate-limiting enzyme is the 7-alpha hydroxylase with the enhancer as the N-acetyl-glutamate. In the urea cycle, the rate-limiting enzyme is the carbamyl phosphate synthetase 1 (CPS1) with the regulator or enhancer as the N-acetyl-glutamate. In the porphyrin or the heme synthesis, the rate-limiting enzyme is the delta-aminolevulinic acid synthase or the ALA synthase, and the inhibitor is the heme. In the pyrimidine synthesis pathway, the rate-limiting enzyme is the carbamyl phosphate synthetase 2 (CPS2) with the regulator or enhancer as the ATP and the PRPP, that is phosphoribosyl pyrophosphate, and the inhibitor is the uridine triphosphate. In the purine nucleotide synthesis, the rate-limiting enzyme is the phosphoribosyl pyrophosphate glutamate aminotransferase enzyme, and the inhibitors are the AMP, ionosine monophosphate, and the guanosine monophosphate. In the purine catabolism or the uric acid synthesis, the rate-limiting enzyme is the xanthine oxidase. So, that's all for today. I hope you found this helpful. Thank you so much.